So, good evening, all. Today we are going to start week 9 lecture series of host pathogen interaction. So, I'm Ananya Chatterjee. I'm a graduate student in IISc Bangalore, and I am one of the teaching assistants of the course Host Pathogen Interaction. Course instructor is Professor Himanshu Kumar from Department of Biological Science, IISER Bhopal. So, we will start with discussing assignment. So the first question is, which of the following is not a characteristic of antigens? Option 1, molecular size. Option 2, chemical composition. Option 3, immunogenicity. Option 4, solubility. Option 5, foreignness. Which of the following is not a characteristic of antigens? So, so the question is which of the following is not a characteristic feature of antigens? So the answer will be solubility. Okay. Which of the following statements regarding B cells and antibodies is false? Option 1, B cell matures in the bone marrow. Option 2, antibodies are also known as immunoglobulins. Memory B cells provide long-term immunity. B cells directly kill infected cells. Antibodies can neutralize pathogens. So, which of the following statements regarding B cells and antibodies is false? Sorry. So, the answer will be B cells directly kill infected cells. This statement is not true. Which of the following is not a type of T cell? 1, helper T cell, killer T cell, memory T cell, plasma cell, regulatory T cells. So the answer is plasma cells, because plasma cells is not a type of T cells, rather it's a type of B cell. What is the role of antigen presenting cells in T cell activation? Secrete cytokines to stimulate T cell proliferation, directly attack pathogens, present antigens to T cells via MHC molecules, produce antibodies against antigens, inhibit T cell activation. So the question is, what is the role of antigen presenting cells in T cell activation? So those antigen presenting cells, what they do, they present antigens, they process those antigens, and then they present those through MHC molecules to T cell. Okay. Which enzyme is commonly used to digest antibodies into Fab and FC fragments? The enzyme is papain. The enzyme is papain. So papain is commonly used to digest antibodies into Fabs and FC fragments. Okay. Next question. Which part of the antibody more specifically binds to antigens? Variable region, constant region, FC region, Fab region, light chain. So they have stated that which part of the antibody more specifically. So we have to say that it's variable region, because Fab region also consists of, like, you know, Fab region is also like, in the Fab region there is also variable region, but if you need to say very much specifically, you have to say that it's variable region, not a constant region. Okay, Fab means fragment of antibody binding region. Okay, FC means constant uh, region, fragment of constant region, and light chain and constant region. These are the options. Answer will be variable region. Which of the following antibody types is typically found in secretions such as tears, saliva, and breast milk? So, which of the following antibody types is typically found? So, we all know that IgA is typically found on in this secretions such as tears, saliva, and breast milk. Which of the following is or are correct? Incorrect. Option 1, hapten is too small and lacks immunogenicity. If hapten is coupled to a carrier molecule, that is protein, an immune response can be induced. Polymers composed of multiple copies of the same amino acid or sugar tend to be strong immunogens. So, which one is incorrect? Polymers composed of multiple copies of the same amino acid or sugar tend to be strong immunogens. No, this is false. Which among the following statements is or are incorrect? Bovine serum albumin BSA is a poor immunogen. Poor immunogen size will be greater than 10,000 Daltons. Sperm specific antigens are immunogenic in nature. Okay, so option 1 and 2 will be incorrect. These are not true statement. Which among the following statements are incorrect? B cell epitopes tend to be on the outside of the antigen. Upon immunization with glucagon hormone, B cell and T cell epitopes won't differ. Monoclonal antibodies are used in pathogen detection applications. Which among the following statements are incorrect? So, B cell epitopes tend to be outside of the antigen. True. Upon immunization with glucagon hormone, B cell and T cell epitopes won't differ. It will differ. The option is incorrect. Monoclonal antibodies are used in pathogen detection application. This is correct. So answer will be option 2 only. So now we will talk about, we'll start about viruses and immunity associated with viruses. So I am now following one book, Principles of Virology by Flint. Okay, the part 1, the science of virology. It consists of foundations and the infectious cycle. So we all know we all know that why we study viruses. We have faced a massive pandemic in 2019 to 2021. So we know that viruses are all around us, comprising an enormous properties like proportions of our environment in both number and mass. So they, like, we inhale a lot of viruses every minute. They enter our digestive systems with the food we eat. They are transferred to our eyes, mouth, and other points of entry. Our bodies are kind of reservoirs for viruses that reside in our receptory, like respiratory organ, gastrointestinal, urogenital tract. In addition to viruses that can infect us, our intestinal tracts are loaded with myriad plant and insect viruses, as well as hundreds of bacterial species that harbor their own constellations of viruses. Viruses can cause human diseases. So, with such constant exposure, it is nothing short of amazing that the vast majority of viruses that infect us have little or no impact on our health or well-being. So, we owe such relative safety to our elaborate immune defense system. We have evolved to fight microbial infection. So when our defense systems gets compromised, even the most common infection can be lethal. Despite such defenses, some of the most devastating human diseases have been or still are caused by viruses. These diseases include smallpox, yellow fever, poliomyelitis, influenza, measles, and AIDS. Viral infection can lead to life threatening diseases that impact virtually all organs, including the lung, liver, central nervous system, and intestine. Viruses are responsible for approximately 20% of the human cancer burden and viral infections of the respiratory and gastrointestinal tracts. Viruses infect all living things. So, if we discuss about some basic important things, that the field of virology encompasses viral discovery, the study of virus structure and reproduction. Viruses are not solely pathogenic nuisance, they can be beneficial. Viruses contribute to ecological homeostasis, keep our immune responses activated and alert, and can be used as molecular flashlight to illuminate cellular processes. Viruses have been part of all of human history. They were present long before Homo sapiens evolved, and the majority of human infections were likely acquired from other animals. Viruses are obligate intracellular parasites and depend on their host cell for all aspects of viral life cycle. While Koch's postulates were essential for defining many agents of diseases, not all pathogenic viruses fulfill this criteria. And viruses can be catalogued based on their appearance, the host they infect, and the nature of their nucleic acid. And also viruses can be beneficial as well. There are some points despite the appalling statistic from human and agricultural pandemics or epidemics, it is important to realize that viruses can also be beneficial. Such benefit can seen most clearly in marine ecology, where virus particles are the most abundant biological entities. Indeed, they comprise 94% of the nucleic acid containing particles in the oceans and are 15 times more abundant than the bacteria and archaea. So, this are characteristic smallpox lesions in a young victim. You can see this illustrations. These are because of the smallpox. These blisters are created. So, what is the origin of vaccinia virus? Over the years, at least three hypotheses have been advanced to explain the curious substitution of cowpox virus by vaccinia virus. Recombination of cowpox virus with smallpox virus after variolation of humans, recombination between cowpox virus and animal pox viruses during passage in various animals, genetic drift of cowpox virus after repeated passage in humans and animals. So, microorganisms are pathogenic agents. So these are the different electron micrographs of virus particles following negative staining. So this is a electron microscope images which is negatively stained, mostly stained with a stain which is called uranyl acetate. Okay, so this is a complex non-enveloped bacteriophage. This is the helical non-enveloped particle of tobacco mosaic virus. C is enveloped particles of the rhabdovirus, vesicular stomatitis virus, and D is non-enveloped icosideral human rotavirus. Simple infectious cycle of a virus is the first step is attachment and entry. So virus is attaching with the cell membrane and then enter. So this is a viral infectious cycle. The infectious cycle of poliovirus is shown as an example. So where after entering you can see that it is releasing its genome, and then the genome gets translated, and then replication is happening, protein production is happening, and then it's getting packaged in a full variant and is getting released. So in our virology experiments, what do? We propagate viruses in different mammalian or mosquito cells. So these are different kinds of cell we use to do culturing viruses. This is a confluent cell monolayers photographed by low power light microscope. So here we can see that this is a primary human foreskin fibroblast. This is mouse fibroblast or 3T3 cell line, and C is continuous line of HeLa cell line. So influenza virus is a highly variable virus. These viruses keep on changing, and due to the property of influenza virus, we always have a some or other epidemic all over the world. And probably you were very well about 100 years back, this was a massive pandemic and that killed many people. So we will have several sessions about influenza virus, and towards the end we'll learn what kind of disease, how they infect the host cell, what are the target cells, what are the host they are infecting. So as you know that influenza virus caused Spanish flu in around 1918. Since 1918 it was there for several years, and this Spanish flu, as you can see, because the most severe pandemic. So when this epidemic becomes bigger size, and far more bigger size means they cross the continents and several continents. So the virus, basically influenza virus, they are present in almost all living animals. Here you can see this can infect like uh, from a bird which stays in like remains in a water body. It can propagate through different animals which um, states or remains in different animals, small animals in the land. So let us look at the genome of influenza A virus. So you can see that there is PB2, PB1, PA. So PB2, PB1 and PA, this makes a complex which is having the RNA polymerase activity, and this RNA polymerase activity is needed in order to make more copies of influenza virus. So try to understand this RNA virus, which is not a base pair, and it has 2341 bases. So there is NP protein that consists of 1565 bases, and it has a role in RNA binding, RNA synthesis, RNP nuclear import. Another protein is Na, which is Na is neuraminidase. This is a surface glycoprotein and it has a neuraminidase activity, and this protein or this enzyme activity is needed for the release of virus after infection. If neuraminidase activity is inhibited, then we can control the virus infection. There is also a protein which is called M1 protein, which is basically consists of 1027 bases. The M1 protein is basically a matrix protein. There is M2 protein which plays an important role. Basically this is a membrane protein and this has ion channel activity. It takes the proton and plays important role in assembly and binding. So in it has say 8 segments in number, and if you join the segments, the total size of the nucleic acids will be around 13.6 kilobases. You can see the structure, and the inside this virus you can see the genome: PB2, PB1, PA, HA, NP, NA, MNNS. M1 protein plays an important role in virus assembly. M2 protein plays an important role in uncoating. They take the proton and then the virus gets dismantled inside the cell. Na, this is playing very important role in virus budding off. This is an enzyme and is a neuraminidase. The NP protein is basically providing an internal framework in order to when all this genome is packing, and then providing kind of framework. HA protein is very important in attachment with the target cell. Okay. So the basic of influenza viruses. So in this previous lecture, it was discussed about the structure of influenza virus, and uh, you have seen that there are four types of influenza virus. The A type basically infect human and has a pandemic potential. So A type is the most dangerous, so they can infect human and they can cause pandemic. And this A type is consist of 8 segments of genome, as you have seen in our previous session. And another is B type. B type basically infect human and has endemic potential. They have endemic, they are not so dangerous. Epidemic potential but cannot infect animal and can cause epidemic, and this B type also has 8 segments. Another is C type. C type is basically causes mild illness to human, lack neuraminidase enzyme, and this lacks Na, and therefore this has a only 7 genomic segment. So, naming system of influenza is basically it is initiated with antigenic type like A, B, C, D, and their antigenic type. The naming includes the host origin. Host origin means if the virus is detected from pig, then that is a host origin, and the first detection like pig, like horse, chicken. If it's a human host, then we do not write, we just write A. So here you can see the example. So virus originated from duck, it's called avian influenza A, and you can see A is written as capital A, can infect the human, and it's written duck. Another example, the human examples, seasonal influenza A, H3N2. So for influenza A virus, the hemagglutinin HA and neuraminidase Na antigen description are also provided in bracket. Here you can see influenza A H1N1 is well written. So H1 and N1 is hemagglutinin, and N stands for neuraminidase. So it is very interesting to understand the hemagglutinin protein. The HA hemagglutinin is which is basically interacting with the host molecule known as sialic acid. It interacts with sialic acid, and then this will trigger the binding of this virus with target cell, and then there will be this HA protein plays a very important role in receptor binding as well as fusion. Membrane fusion is taking place, and then this ribonucleoproteins are transported inside cells. So their major function is a receptor binding and membrane fusion, but they may also have some structural roles for HA in budding of after a virus replication. There is a release of virus and egress of virus, so they may play important role in budding of virus particle formation. How influenza virus and infect human and human cells. Basically, it is started with the cleavage. There is a cleavage site present in HA, which is varies between viral strain to strain, and can affect the so if So basically, these enzymes are basically created or cleaved this HA protein in this H1N1, the Spanish flu virus, and has a monovesic cleavage motif. The HA is very important antigenic determinant, and if we mask this HA by some monoclonal antibody or by other mean, then we can prevent the infection. So we will now talk about the proteins which are present on the membrane of influenza viruses. So this is a membrane protein which is present in the membrane of influenza viruses, where you can see there are receptor binding sites. These are different antigenic sites, and then it has fusion protein. So it's the protein is folded in at a neutral is like that at neutral pH, but at fusion pH it gets folded back to straight positions, and then where you can see the fusion peptide membrane anchor and then globular head, and then it helps in fusing of the membranes, viral membranes and the endosomal membrane. So, what is the mechanism of entry? Influenza viruses require a low pH to initiate fusion, and therefore internalized by endocytic compartments. There are four internalization mechanisms: via clathrin coated pits, via cavuli through non-cavuli pathways through macropinocytosis. The uncoating of influenza viruses in endosomes is blocked by changes in pH caused by weak bases. Ionophores effective uncoating is also dependent on the presence of M2 protein. Amantadine and romantadine inhibit replication immediately following virus infection. Pathogenesis in humans: infect superficial cells of the respiratory tract, alveolar macrophages and dendritic cells can be infected. The site of optimal growth in the respiratory tract for influenza viruses determined by the prevalence of Ca alpha 2 3 gal or Ca alpha 2 6 gal receptors. So reverse genetic system for generating IAV or influenza A virus. Each plasmid system bidirectional promoters are there. So this is virus PR8 H1N1 vaccine strain or wild type strain. So this is cell culture HIC 293 plus MD CK culture. So we can also generate different influenza strains with PR8 with PB1 deficient in microRNA 485 target site. Is flu and cold is same. So what happens in the flu or cold, what are the symptoms? Fever, headache, tiredness, body aches, runny stuffy nose, coughing and sneezing, sore throat. 50% of cold is caused by rhinoviruses. It's also caused by coronaviruses, respiratory syncytial viruses. Flu or common cold is caused by influenza viruses. So influenza it comes from an Italian word meaning influence, and flu means flu came from like from the word influence. Flu is came from the word influence. Okay. So clinical features: asymptomatic infection to primary viral pneumonia that rapidly progresses to a fatal outcome. Symptoms: abrupt with headache, chills, dry cough, which are rapidly followed by high fever, malaise, anorexia. Substernal tightness and soreness can accompany the cough. Prominent sign of infection is fever that often peaks within 24 hours at 38 degrees Celsius to 40 degrees Celsius. The elderly can have high fever, lassitude and confusion. Conjunctival inflammation and excessive tearing may occur. Illness is more frequent and more severe in cigarette smokes. In children, viruses can be found for up to 13 days after the onset of symptoms. In immunocompetent patients, the PDM09 virus is typically shed from the day prior to the onset of symptoms to 5 to 7 days after the onset of illness. Influenza virus and disease outcomes in different hosts. So H5N1 or influenza virus, natural reservoir or host is duck. Viral infection, asymptomatic infection, immune response, low cytokine levels and high viral recognition. In transmission host, mild to severe infection, some increased cytokine production. And for spillover host or terminal host, this is severe infection, high levels of cytokines, chemokines and acute phase proteins produce, or this is also called cytokine storm. Infection induces changes throughout the respiratory tract, but the most clinical important pathology develops in the lower respiratory tract. Acute diffuse inflammation of the larynx, trachea, bronchi are observed with mucosal inflammation. So influenza A virus infection has been shown to exacerbate asthma. Children, especially those younger than 3 years, have a higher incidence of GI tract infections such as vomiting and abdominal pain. So who are vulnerable to flu? The pregnant women, people with chronic medical condition, elderly people, healthcare workers, and children under 5 years. So now we will discuss about arbovirus infection. So arbovirus, that is also called arthropod-borne viral diseases. So arthropod-borne pathogens account for more than 17% of infectious disease. Blood-feeding arthropods such as mosquitoes, ticks, fleas, kissing bugs, biting flies serve as biological factors. Blood-sucking arthropods transmit virus from one vertebrate to another viremic host. Most arboviral diseases are caused by infected mosquitoes. Several hundreds arbovirus are known, however, approx 100 viruses are known to cause disease. The most prevalent arboviral diseases per year are dengue virus, chikungunya virus, Zika virus, yellow fever virus, Japanese encephalitis virus, and West Nile viruses. Most of the arboviral mosquitoes are the villain in carrying these pathogens, and most of these diseases are caused by mosquitoes. So as we were stating, like Zika virus, myrovirus, yellow fever virus, dengue virus, and so on. Ticks can also spread the virus. Tick bites can cause disease like tick-borne encephalitis. So, what makes us attracted to mosquitoes? Body odor. So, certain compounds like ammonia, carbon dioxide present in our breath, skin, and sweat attract mosquitoes. Skin bacteria. The high diversity of microbes on skin may be less attractive to mosquitoes. Pregnant women may be more attractive to mosquitoes due to body temperature increment. General characteristics of parvovirus infection: fever with or without benign encephalitis, that is inflammation of brain causing high fatality. Hemorrhagic fever, it is quite severe and fatal. General pathogenesis and pathology. Virus multiply in myeloid, lymphoid, and vascular endothelial cells. If virus cross blood-brain barrier, then it multiply in nerve cells of CNS and may cause encephalitis. Neuroinvasion depends on the level of viremia, genetic background, innate and adaptive immune status. Arbovirus infections show age dependent susceptibility to CNS. Incubation period is 4 to days, causing flu like illness to encephalitis. Some individuals show drowsiness with stuporous, completely suspended sense or sensibility. General diagnosis: recovery of virus, direct detection. Immunity against arboviruses: permanent humoral and cellular immunity develop after infection. Treatment: personal protective measures, protective clothes, insect repellents. Most of arboviral disease treatment is symptom management. So you can see the dengue statistics, the global evidence consists risk and burden of dengue. So the green color is complete absence. Then the dark red is complete dominance or complete presence. So dengue virus is a member of flavivirus genus of Flaviviridae family. Dengue contains single-stranded positive sense RNA. It can cause a full spectrum of clinical manifestation following dengue infection, ranging from asymptomatic infection to dengue fever. Sorry. And the most severe disease, dengue, hemorrhagic fever, and dengue shock syndrome. Aedes aegypti is the main vector for dengue transmission to humans. Aedes albopictus can also sustain transmission. About 1 in 4 people infected with dengue will get sick. About 1 in 20 people will get sick with dengue, will develop severe dengue. Transmission of dengue viruses. So, sylvatic or enzoonotic, where from Aedes mosquito it will run through primates, and then mosquito again it will be circle, but also it from a mosquito it can get go to human, and then it will become a endemic. Different stages of dengue virus. So dengue fever, febrile illness with 2 or more of the following like headache, retroorbital pain, myalgia, arthralgia. Dengue hemorrhagic fever, fever 2 to 7 days, and then bruising, increased vascular permeability, thrombocytopenia. Dengue shock syndrome, rapid or weak pulse, narrow pulse pressure or manifestation, time course of clinical signs and symptoms. So in between 4 to 12 days there will be viremia, headache, pain, fever, rash, and following days there can be bruising, thrombocytopenia, and shock syndromes. Different serotypes of dengue. So based on the neutralization assay, 4 genetically similar but antigenetically distinct serotypes of dengue was identified: 1, 2, 3, 4. Infection with one serotype of dengue confers lifetime immunity against that serotype but not against heterologous serotypes. Secondary infection with a heterologous serotype can lead to severe complications like dengue shocking syndrome, and due to the process called antibody dependent enhancement. So 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 what is antibody dependent enhancement? It's like that whenever you have some antibodies present in your body and one strain of dengue is infecting, so but those antibodies which are present in your blood, those were generated well due to the previous infection of other serotype. But now those antibody will bind to the new serotype, but what will happen is that they won't be able to get neutralized completely. So those FAVs will get recognized by Fc gamma receptors which are present in different immune cells like monocytes, and then those viruses are then gets engulfed inside the cell and then produces a lot more viruses, hence increases viral load. So what is dengue virus life cycle? So like mosquito infection happens, mosquito takes a blood meal from a person with acute dengue, and then extrinsic incubation. So what happens, that is virus infects the midgut, eventually travels to the salivary gland, and inside this mosquito it takes around 8 to 10 days. And human infection, so one mosquito can infect several humans. Intrinsic incubation, the onset of symptoms usually take 4 to 7 days. Factors involved in the control of dengue virus disease response: immune status, viral strain, genetic status, age. Dengue diagnosis, so acute illness at the very first few days, and then the viremia, the load of virus is also high, and then after that the soluble antibodies are getting increased. Okay. Dengue treatment and prevention: no specific treatment for dengue is currently available. Analgesic and antibiotic drugs such as paracetamol. Fluid therapy is a key to dengue management. Prophylactic platelet transfusions are given for dengue fever with thrombocytopenia to prevent hemorrhagic complications. Arbovirus infection or Zika virus infection. Zika virus is a positive strain RNA flavivirus in the family Flaviviridae. Zika was first isolated in 1947 in the Zika forest in Uganda, where transmission of the ancestral African lineage of Zika virus was limited to the enzootic transcirculation between non-human primates and sylvatic Aedes mosquitoes with sporadic spillover. Zika is also spread mostly by the bite of infected Aedes mosquito. So this is congenital Zika syndrome. Clinical manifestations: severe microcephaly, collapsed skull, redundant skull, skin, hip dislocations, development delay. Zika virus associated Guillain-Barré syndrome. So Zika virus associated Guillain-Barré syndrome is estimated to be 2 to 3 cases per 10,000 ZB infections. Acute inflammatory demyelinating polyneuropathy, acute motor axonal neuropathy, and the Miller-Fisher syndrome have been observed with Zigbee associated Guillain-Barré syndrome. Introduction to bacterial infection. Bacterial infection, intracellular or extracellular. So intracellular means Listeria. Example of extracellular: Salmonella, Shigella. Route of entry: broken skin, Clostridium tetani causing tetanus, respiratory tract infection. Infection to the host and disease outcome depends on inoculum size, virulence of bacteria. Immunity against bacterial infection. Innate immunity against bacterial infection by humoral components. Innate immunity against bacterial infection by cellular components. For extracellular bacterial infection, antibody and Th2 immunity for extracellular bacterial infection via complement activation. The infection process: attachment or adherence to host cells, that is epithelial cells, multiply and spread tissues or via the lymphatic system. Genomics and bacterial pathogenicity. The clonal nature of bacterial pathogens. There are many types of Haemophilus influenza, but only clonal influenza B type B is commonly associated with disease. Mobile genetic elements. Primary mechanisms for exchange of genetic information between bacteria include natural transformation and transmission mobile genetic elements such as plasmids, transposons, and bacteriophages. Pathogenicity islands, large groups of genes that are associated with pathogenicity are located on bacterial chromosomes are termed pathogenicity islands. So examples of virulence factor encoded by genes on mobile genetic element. So genes, genus and species plasmid encoded E. coli virulence factor and disease, heat labile and heat stable enterotoxins that cause diarrhea. Virulence factors during bacterial infections. Regulation of bacterial virulence factors. Pathogenic bacteria and other pathogens have adapted both to saprophytic or free living states, possibly environments. They have evolved a complex signal transduction system to regulate the genes. Environmental signals often control the expression of virulence genes. Adherence factor. Bacteria must adhere to cells of a tissue surface. If they did not adhere, they would be swept away by mucus or other fluids that bathe the tissue surface. Several factors that play important roles, including surface hydrophobicity and net surface charge, binding molecule on bacteria, and host cell receptor interactions. Bacteria and host cells commonly have net negative surface charges and therefore repulsive electrostatic forces. These forces are overcome by hydrophobic and other mode specific interactions between bacteria and host cell. Invasion of host cells and tissues. For many disease causing bacteria, invasion of the host epithelium is central to the infectious process. Some bacteria, example Salmonella species, can invade tissues through the junctions between epithelial cells. So the last thing we will discuss about characteristics of exotoxins and endotoxin. So exotoxins are excreted by living cells, high concentration in liquid media, and endotoxin integral part of the cell wall of gram-negative bacteria. Exotoxin produced by both gram-positive and gram-negative bacteria. Endotoxin found only in gram-negative bacteria. Exotoxins relatively unstable, toxicity often destroyed rapidly by heating. And endotoxin relatively stable. Withstand heating temperatures above 60 degree. Exotoxin highly toxic, fatal to animals in microgram quantities or less. Endotoxin moderately toxic, fatal to fatal for animals in tens to hundreds of micrograms. Endotoxin usually do not produce fever. Exotoxin and endotoxin usually produce fever in the body. So we will discuss about lecture 10 in the next coming week on Tuesday from 6 to 8 p.m. Thank you all. Thank you for attending the class.